We are starting with classification of egg. On the basis of quantity of yolk present in the egg, the eggs can be classified into three main categories. Microlacithal eggs have a very small amount of yolk present inside them. In case of mesolacithal eggs, we have moderate amount of yolk. In case of macrolacithal eggs, we have a large amount of yolk present. If you see, in case of microlacithal eggs, there is very small amount of dark granules present and they are uniformly distributed all along the cytoplasm. In case of mesolacithal eggs, as you can see this dark region present here, okay, means there is moderate amount of yolk present and distended to one corner of the particular egg. In case of macrolacithal egg, there is huge amount of yolk present and only a very few amount of cytoplasm is clearly visible, as you can see. Now, we will talk about some examples according to this classification. In case of microlacithal eggs, the most common examples would be amphioxus and echinoderms. In case of mesolacithal eggs, the frog eggs are most common example. I will show you the image in upcoming slides. In case of macrolacithal eggs, the most common examples we have seen in our households is the eggs of birds, the eggs of hen. Most of the eggs of reptiles and monotreme mammals are of this macrolacithal nature. Now, when it comes to mammals and especially humans, there is a divide in, or we can say there is a difference in opinion. In case of humans, the eggs are practically alacithal, means there is practically no yolk present inside the egg. All you can see is clear cytoplasm. Very small amount of yolk might be present in mammalian and human eggs at certain stages of development. So, mammalian or human eggs can be placed under this microlacithal eggs or alacithal eggs. On the basis of distribution of yolk, eggs can be classified into again three categories. First is isolacithal eggs, in which the yolk is distributed uniformly all along the cytoplasm, and isolacithal is common with microlacithal eggs, that a very small amount of yolk is present and it is uniformly distributed all along the cytoplasm. In case of telolacithal eggs, it is usually present with the mesolacithal or macrolacithal eggs, and this yolk is usually localized to one end or one corner of the egg. As you can see, this is mesotelolacithal egg, in which yolk is highly concentrated and only restricted to the vegetal pole of this egg. In case of centrolacithal egg, the yolk is restricted to center of this egg and it is surrounded all along by this cytoplasm. If we combine this classification, in case of alacithal eggs, amount of yolk is practically absent and distribution is not considered. Example is humans and mammals. In case of microlacithal eggs, microlacithal is common with isolacithal, so yolk is very little and evenly distributed. Examples amphioxus and echinoderms, as we talked about. In case of mesotelolacithal eggs, the amount of yolk is moderate and usually restricted to vegetal pool. Example is frog and amphibians. In case of macrotelolacithal eggs, the amount of yolk is very high and usually restricted to vegetal pool. It is present in reptiles, in birds, and for monotreme class of mammals. In case of centrolacithal eggs, amount of yolk is abundant but is restricted to center of the egg, and it is usually present in insects. Let's talk about another classification on the basis of presence or absence of shell. If a hardened shell is present, the eggs are known as cladoic eggs, and we can see these types of eggs in most of the birds, most of the reptiles and monotreme mammals. As you can see, European starling bird eggs in a nest. In case of non-cladoic eggs, they are present without the shell. It is usually seen in amphibians like frog eggs, you can see in this picture. They are present in amphibians, maybe in fishes, in mammals, but not in monotremes. Now, what is the composition of yolk? So as you all know, yolk acts as a nutritive material stored in the cytoplasm of the egg. There are proteins, especially the phosphoproteins like phosphatin, lipoproteins, some vitellogenins, lipids. There are abundant amount of triglycerides, phospholipids and cholesterol present, and these lipids act as primary energy source. There are many carbohydrates like glycogen, which provides energy as well. Water is present for specific medium for metabolic activity. There are specific ions and minerals like calcium, magnesium, potassium and sodium, and some other trace elements are also present which will help in enzymatic actions. There may be some kind of pigments like carotenoids, biliverdin in case of amphibian eggs, which helps these amphibian eggs for their camouflage, because amphibian eggs are placed in water itself, as you can see right here. They help in camouflage, sometimes in photo protection as well. What is the significance of yolk? Yolk is a primary nutritive material which is main source of energy and raw material for embryo until its feeding and nutrition mechanism is replaced by placenta, which is present in higher mammals. It also helps in regulation of cleavage. I will make a separate video on cleavage as well. We will talk about how cleavage is decided by the type of egg. It also helps in access determination. Like we said, that yolk may be restricted to one specific end, like it may be restricted to vegetal end, and opposite to which lies the animal end of that particular egg. So it helps in access determination. It has evolutionary significance, which correlates with the reproductive strategy, whether the organism is oviparous or viviparous in its reproductive strategy. At last, we will talk about synthesis of yolk. Yolk is synthesized mostly by maternal contribution, we can say. So there are two main sources of synthesis of yolk. First is endogenous, in which oocyte and its surrounding cells helps in synthesis of yolk. The molecules like glycogen, small proteins and some lipids are contributed by this mechanism. Another one is exogenous mechanism, which is primary source, in which the extra ovarian tissues like liver tissue in vertebrates or fat body in case of insects are responsible for making proteins and stuff like vitellogenin, which is later transported to ovary via blood or hemolymph. So these are two main sources of synthesis of yolk. The synthesis occurs mostly by exogenous pathway and some endogenous contribution as well. And later the secretions are carried out to ovarian tissue through blood in vertebrates and hemolymph in case of insects. In case of vertebrates, the estrogen is the main hormone responsible for this stimulation. Estrogen stimulates hepatocytes, which are liver cells, to secrete and make vitellogenin, and this is transported through blood to ovarian tissue. In case of insects, juvenile hormone and ecdysteroids help the fat bodies to secrete the vitellogenin, and it is transported to ovary through hemolymph. Then in case of oocyte, there is endocytosis, or specifically we can say that there is receptor mediated endocytosis of vitellogenin in ooplasm. The plasma membrane of ooplasm is having vitellogenin specific receptors, which is a type of LDL receptors, and when uptake occurs, the vesicle fusion will take place and receptors will be recycled back. That's how the uptake of this vitellogenin takes place in case of oocyte. Then the processing takes place within the oocyte. This vitellogenin is cleaved by some proteolytic enzymes and it is converted to major yolk proteins like lipobutylin or phosphatin. Lipobutylin, if you know, is lipoprotein and it acts as main energy source. In case of phosphatin, it is phosphoprotein and it binds to ions like calcium and magnesium. The accumulation of crystalline yolk platelets takes place, and depending upon egg, the distribution may vary, and additional storage material is synthesized within the oocyte, which may include the glycogen granules which are contributed by this endogenous pathway, and some lipid droplets as well. So that's all about synthesis of yolk.